Imagine opening a sealed vault that hasn't been touched since the days of the American Revolution. Inside isn't gold or silver, but something far more valuable for understanding who we are. The untouched genetic blueprints of the people who actually built the foundations of the United States. For decades, we have assumed that the millions of Americans who claim Quaker ancestry today are the direct, biological descendants of those quiet, plain-dressed pioneers who settled Pennsylvania and New Jersey in the 1700s. We thought we knew exactly who they were. But a groundbreaking new study has just turned American genealogy upside down. Molecular biologists recently gained access to a series of forgotten 18th-century burial sites. And when they sequenced the DNA from those original Quaker settlers, the results were an absolute bombshell. It turns out that the people we call Quakers today have almost no genetic overlap with the people buried in those 1700s graves. The Quaker lineage that so many families pride themselves on might be one of the greatest biological illusions in history. Today we are peeling back the layers of a genetic mystery that suggests an entire population didn't just move, they were effectively replaced. To understand why this is such a shock, you have to realize that the Quakers, or the Religious Society of Friends, were one of the most meticulously documented groups in human history. They were obsessed with records. They kept marriage certificates, birth ledgers, and migration papers that are still used by genealogists today as the gold standard of evidence. Because of this, the Quaker community was thought to be a closed genetic loop, similar to the Amish or the Mennonites. We expected to see a very specific founder effect in their DNA, a clear line leading back to small villages in northern England and Wales. But when the scientists looked at the ancient samples, they didn't find a unified group of English farmers. They found a genetic signature that was far more diverse, far more complex, and almost entirely absent from the modern American Quaker population. The first clue that something was wrong came from the mitochondrial DNA, the markers passed down from mothers to their children. In the modern population that identifies as having Quaker heritage, you see a very standard Western European profile. It's what you'd expect from people whose ancestors moved from the United Kingdom to the American colonies. But the DNA from the 1700s graves showed a high concentration of rare haplogroups that were common in Scandinavia and the Mediterranean coasts during the Middle Ages, but which almost vanished from the Quaker record by the mid-1800s. It's as if a specific biological group arrived in America, lived for 3 generations, and then simply evaporated from the gene pool. This leads us to the Great Quaker Disconnect. Historians have long talked about the separations of the 1800s, where the Quaker Church split into different factions like the Hicksites and the Orthodox. We always viewed these as religious arguments over theology, but the DNA evidence suggests these splits were actually biological. The group that stayed in the church and kept the Quaker label actually had very little to do with the original pioneers. The descendants of the founding settlers appear to have left the faith entirely, married into the general population, and lost their identity, while a completely different group of converts moved in and took over the Quaker name. We aren't just looking at a change in belief. We are looking at a total demographic turnover that was hidden by the fact that everyone kept using the same label. Think about what this means for the millions of people who have used DNA testing kits to find their Quaker roots. Most of these tests compare your spit sample to a reference panel of people who call themselves Quakers today. But if the modern reference panel doesn't match the original founders, then every single one of those ancestry hits is based on a false premise. You might have a genetic match with a modern Quaker from Ohio, but that doesn't mean you have a drop of blood from the people who arrived with William Penn. The science shows that the modern Quaker profile is largely a Victorian-era construct, a mix of later European immigrants who adopted the Quaker lifestyle long after the original genetic pioneers had faded away. The study went even deeper, looking at autosomal DNA, the chunks of code that tell us about long-term population movements. The original 1700s Quakers showed signs of extreme endogamy, meaning they were marrying within a very tight, specific group for centuries before they even left England. This created a unique genetic signature that should be easy to spot in their descendants. It's like a biological thumbprint. When scientists searched the massive databases of modern DNA companies for that specific thumbprint, they found it in the most unexpected places. It wasn't in the suburban families in Pennsylvania or the quiet meetings in Richmond, Indiana. Instead, it was scattered across the rural South and the deep Appalachian Mountains among people who have no idea they have Quaker ancestry. It seems the true Quakers didn't disappear. They moved into the wilderness, changed their names, and forgot their history. Why would a group so famous for their record-keeping just vanish? Genetics tells us a story that the history books missed. The original Quaker pioneers were survivors of intense social pressure in England. When they got to America, they were highly successful, but they also faced a massive demographic squeeze because they were so strict about marrying out, meaning you were kicked out of the church if you married a non-Quaker. The original genetic pool began to shrink. The scientists believe that the founder DNA we see in the 17th century graves was eventually bred out of the formal Quaker organization. Those who wanted to marry for love or for land left the church, taking their unique DNA with them into the general American melting pot. Meanwhile, the formal organization was replenished by new converts from different parts of Europe who had no biological connection to the original group. This creates a surreal reality where the people who look and act like Quakers today are the biological imposters, and the real Quakers are living in the mountains of North Carolina or the plains of Texas, thinking they are Scots-Irish or German. It is a total reversal of family lore. If your family has a tradition of being Quaker for 300 years, the DNA suggests that somewhere along the line, your ancestors were actually integrated into the name rather than born into the bloodline. The researchers also found evidence of hidden gene flow in the 1700 samples that challenges our understanding of colonial life. In several of the earliest Quaker graves, they found genetic markers that were clearly non-European. This is a massive revelation because Quakers were among the first to formally oppose the practice of holding others in bondage, but it was always assumed they remained a strictly European-descended group. The DNA tells a different story. It shows that the early Society of Friends was a much more inclusive biological community than we ever imagined. There was significant intermarrying and integration happening on a genetic level that was never written down in the official meeting minutes. Over time, as the movement became more respectable and middle class in the 1800s, these diverse lineages were purged or ignored, leading to the more homogenized Western European Quaker look we see today. This brings us to the genetic erasure of the 1830s. During this time, the Quaker movement underwent a series of massive internal shifts. The DNA evidence shows a sudden narrowing of the gene pool in the official records right during this period. It's as if the church went through a genetic cleansing, where only certain families were allowed to remain in leadership and pass on the identity. When you compare the DNA from a grave in 1750 to a grave in 1850 in the same cemetery, the difference is night and day. Even though they are buried in the same ground and have the same surnames, they aren't related. The names stayed the same, but the people changed. For anyone who loves genealogy, this is a nightmare scenario. We rely on surnames to track our ancestors. We assume that if your name is Pemberton and your family has always been Quaker, then you are a Pemberton through and through. But the DNA proves that non-paternal events, situations where the biological father isn't the one on the birth certificate, and the adoption of new members into families were rampant. In the Quaker community, the family name was more like a club membership. If a new convert was adopted by a prominent Quaker family, they often took the name and passed it down as if it were their own biological heritage. Over 200 years, this process completely replaced the original genetic stock. The scientists involved in the study used a technique called principal component analysis to map the samples against a global database. The results were visually stunning. The 17th century Quaker samples clustered in a very tight, isolated bubble near the ancient inhabitants of the Danelaw region of England, the area once controlled by Vikings. This suggests the original Quakers were a very specific, northern subgroup of English people, but the modern Americans who claim Quaker ancestry are spread out across the entire map of Europe. They represent a pan-European average, not a specific pioneer group. This proves that the modern Quaker identity is a social construct that has overwritten a biological reality. What does this mean for our understanding of American history? It suggests that the cultural carriers of the Quaker values, the people who actually passed down the ideas of equality, peace, and simplicity, might not be the biological descendants of the people who invented those ideas. We have separated the idea of being a Quaker from the blood of the founders. In a way, this is a beautiful testament to the power of the philosophy. It was so strong that it could be adopted by entirely different groups of people, who then claimed it as their own history. But for the scientists and the genealogists, it is an uncomfortable reminder that our family trees are often works of fiction. The study has also opened up a debate about genetic sovereignty. Do the modern Quaker organizations have the right to claim the history of those 1700 settlers if they aren't actually related to them? If the DNA shows that the true Quakers are actually a group of unassociated people in Appalachia, who does the history belong to? We are entering a new era where ancient DNA can challenge the origin stories of entire religious and social groups. The Pennsylvania story we tell ourselves in school, the story of a continuous line of friends from Penn's Woods to the modern day, is genetically impossible. As the data continues to pour in, the researchers are finding even more anomalies. There are specific genetic diseases that are common in the modern Quaker community, such as certain types of hearing loss and metabolic disorders. We always thought these were Quaker genes passed down from the founders, but the DNA from the 1700s graves shows none of these markers. This means these community diseases actually entered the Quaker population much later, likely from German and Dutch converts in the 19th century. We have been misattributing biological traits to the wrong era for over 100 years. This mismatch also explains why so many people get unexpected results from sites like Ancestry DNA or 23andMe. You might have a family Bible that says your great-great-grandfather was a Quaker from Philadelphia, but your DNA test says you are 60% Irish and 20% Scandinavian. You assume the test is wrong, or that your grandfather was an outlier, but the science suggests your grandfather was the norm. He was part of that massive wave of replacement individuals who took on the Quaker mantle while the original genetic line was being absorbed into the rural hinterlands of America. The lost Quaker DNA is now being found in the most fascinating places. Scientists are looking at the triad ethnic groups of the coastal Carolinas, communities that have long claimed to be a mysterious mix of various backgrounds. When they sampled these groups, they found the exact Danish-English signature of the 17th century Quakers. These people, often marginalized and forgotten by mainstream history, are the true biological heirs of the people who founded the Pennsylvania colony. They didn't fail to keep their heritage. They just kept the blood while the city dwellers kept the name. Think about the irony here. The plain-dressed, silent-meeting Quakers of today, who are seen as the authentic representatives of their history, are genetically disconnected from the source. Meanwhile, someone wearing a trucker hat and living in a trailer in the Blue Ridge Mountains might be the genetic spitting image of the high-born English pioneers who walked with George Fox. It's a total disruption of social class and historical memory. Biology doesn't care about your church membership or how well you keep your records. It only cares about who actually had children with whom. The researchers believe this population replacement happened in 3 major waves. The first was during the American Revolution, when many of the original peace testimony Quakers were forced to flee deep into the frontier to avoid the conflict. They lost touch with the central meetings and their DNA drifted into the general pioneer population. The second wave was the industrial conversion of the 1850s, where thousands of new factory workers in cities like Philadelphia and Birmingham joined the Society of Friends for the Social Benefits, diluting the original gene pool. The third wave was the ecumenical movement of the 20th century, which brought in even more diverse genetic backgrounds. By the time we started doing DNA testing in the 2000s, the original founder's signature was long gone from the official community. This is a mega project of the soul. The building of the American identity was a project that spanned centuries, and the Quakers were one of its most important architects. But just like a skyscraper that is rebuilt so many times that none of the original steel remains, the Quaker identity has been entirely renovated. The DNA from those 1700s graves is the original steel. It's the ground truth of who started this whole experiment. And finding out that the original steel is missing from the modern skyscraper is a realization that is sending shockwaves through every history department in the country. The scientists are now calling for a remapping of American colonial history. They want to move away from using religious labels to describe populations and start using genetic clusters. Instead of saying the Quakers settled this valley, they want to say genotype A settled this valley, and then was replaced by genotype B, who adopted the name Quaker. This is a much more accurate way of looking at how the continent was actually populated. It removes the mythology of the family tree and replaces it with the hard, cold reality of the double helix. If you are one of those people who has a Quaker branch on your family tree, this information might be disappointing. You might feel like your history has been stolen, but there is a different way to look at it. It proves that the American dream was more effective than we ever realized. The original pioneers were so successful at integrating, so successful at bringing people into their worldview, that their own unique biological signature was sacrificed to the larger whole. They didn't die out. They became everyone. They are the ghost ancestors of millions of Americans who will never know their name. 1700s graves are a time capsule, a final look at a group of people before they were diluted into the greatness of the American experiment. The disconnect between that DNA and the modern Quakers isn't a failure of the community. It's a symptom of how dynamic and fluid our history actually is. We like to think of human groups as static, unchanging things. We want the Quakers of today to be the exact same people as the Quakers of 300 years ago. But people aren't museums. We move, we change, we marry, and we reinvent ourselves. The DNA is just the record of that constant motion as we continue to sequence more ancient samples, not just from Quakers, but from the Puritans of Massachusetts, the Cavaliers of Virginia, and the Huguenots of New York. We are likely to find the same thing. The founding DNA of America is likely hidden in places we haven't even looked yet. It's a massive countrywide mystery that is only just beginning to be solved. The American Quaker DNA mismatch is the first major crack in the facade of our genealogical certainty. It's a reminder that while we can keep our family names and our religious traditions for centuries, our blood has a mind of its own. As you look at your own DNA results, remember that you are a mosaic. You aren't a direct descendant of a label. You are the result of thousands of years of people moving, hiding, changing their identities, and surviving. The true Quakers of the 1700s might not be in the church today, but their code is still out there, floating in the sea of American genetics, waiting to be recognized. The science isn't destroying our history. It's just showing us that the history is far more interesting and far more chaotic than we were ever told. The mystery of the 1700s DNA is a call to look closer at who we are and where we actually come from, beyond the names in the leather-bound books.